This is Betsey Dyer. She's the biology. She's a professor at the biology at Wheaton College in Massachusetts. And I think what is interesting to me is she actually wrote a textbook or a field guide to bacteria. She calls herself, able to think in terms of bacteria. And I think maybe the story that I remember was the idea that she's out in Yellowstone and Old Faithful goes off and she's down in the pools looking at the hot streams because there's all kinds of neat stuff going on. She feels like you don't need a microscope to study bacteria. They're all over the place. So I'm really excited about having her here. So thank you.
Great. Great. Thank you. This is a complicated title. You might be able to tell that the first part of the title was actually, I think, something that Adam made up. He was eager for me to talk about the bacteria being our second brain and actually had even a more elaborate thing for me. As you'll get to know me in the next 30 minutes, you'll see that the second part of the title is actually where I tend to go with these sorts of topics. I get pretty specific. So it's about bacteria and it's about neurotransmitters. So if we're going to talk about the brain, I'm going to get down to the neurotransmitters that are the signal molecules that run our brain. And then I'm going to get into some experiments, I hope in a user-friendly sort of way. But I start with some take-home stories. I never assume this for any audience, except that we've just had a beautiful talk by Doug, so maybe I can assume that for this audience, that microbes comprise most of the diversity and abundance of life. And that includes the bacterial symbionts of animals, and we are animals, so we will eventually get to humans in this talk. And here's maybe the big surprise, or one of the big surprises. Bacteria can make neurotransmitters using ancient pathways that have probably been around for billions of years. And I'll try to make that also understandable. That should be a surprise. And that's the gist of this presentation. But, because here's what we really want to know, we don't know much about the consequences of those neurotransmitters that are being produced by symbiotic bacteria of animals, and that's where I'm going to end up going with this within a half an hour. All right, starting with this first statement that I made. I do want to say something about this. I do want to set the scene, the microbial scene here. There is a group at University of Georgia who a number of years ago tried to calculate the total number of bacteria on Earth, and they did it in a Gedanken kind of way, right? They didn't go literally around counting, but they would, for example, get a number of all the bacteria in a cubic meter of ocean water at a certain depth, at a certain place on the Earth, and then do a multiplication problem over the course of an appropriate area, and then take another cubic meter of water, maybe deeper, and do the same thing. Do the same thing with soil at the surface, same thing with soil at a depth. Do the same thing understanding that bacteria are in and on all the other organisms on Earth. So you can see that it was a gigantic mathematics problem that this group at University of Georgia did, and they came up with a number so crazy that we don't have any way of comprehending it. We have a hard enough time just distinguishing a billion and a trillion. They both just sound like really huge numbers. We know one of them is bigger, but how much bigger usually doesn't immediately pop into our head. So it's a ridiculous number, but the take-home story on that is that they are by far the most numerous of all organisms on Earth. And they are the most diverse of all organisms on Earth. This is not supposed to be readable even from the front row, but look how it looks like a phoenix taking off. The people that came up with this family tree of all organisms on Earth, including mammals, I think deliberately made it have this beautiful kind of bird-like look to it. I will say that everything that you see there is some sort of a microbe. We are on it down in the lower right-hand corner. There's a I think this is also the day that we make fun of scientific terminology. Opisthocont, terrible name, but we are opisthocons down there, and not just us. Pretty much everything that we know as an animal is down here in the opisthocons. A horrible name, so don't try to use it or anything. But this is to keep us humble. We're scarcely on the diagram. In fact, humans are not on this diagram. Most of the diversity on the planet, the vast majority, is microbial. That's what this says. And yet they're featureless. I sometimes tell my students, you could put all of the morphologies of all of the bacteria on Earth on a piece of paper using the punctuation keys of your keyboard. Some dots, some dashes, some squiggles, some half parentheses, and you've got it. So they're doing it all with function, not form. They are not about what they look like, they're about what they do. So all that incredible diversity that I showed you in the rising phoenix picture, it's not about what they look like, and that's tough for humans because we're all about form. We're very visual, we're very morphological, and this is not about morphology for the most part. It's about what they do, but what they do is extraordinary. Okay, next take-home story on my short list there. And that includes the bacterial symbionts of animals, also extraordinarily diverse, and yet nothing much to look at for the most part if you get them under a microscope. And I need to do something right now with pathogens just to make sure, and I know this is an audience that's very open-minded about microbes, but just in case you're thinking, well, wait a minute, we're talking about some pathogens here, there's actually something that I recommend to you. Look down at the bottom. Oh, you can't quite see it. I'm going to say it. It's cut off the screen. That's okay. Something called microbe cards that you can buy on Amazon for about $15, invented by someone named Mark Pepler. They are designed for medical students and others who are trying to memorize their pathogens. They are designed like baseball cards. The front cover, and this is the front cover of one, shows you a picture of the microbe, string of little rods, and the back is all the stats on that microbe, just like a baseball card. The mnemonic aid for you, though, remember these are the pathogen cards. The mnemonic aid is that the complete set of bacterial pathogen cards is about 50. So it's about the size of a deck of cards, about 50. And now, think back to what I told you about the sheer number of bacteria on Earth. It's actually an extremely rare thing for a microbe to be a pathogen. Tends to be center in our minds sometimes, but it's actually extremely rare. I think the question about pathogens is not how come you're a pathogen, but how are you even managing to be a pathogen? Apparently, it's not a real sustainable lifestyle for most microbes. It's a very unusual lifestyle. All right, so I've just dismissed pathogens. All right. Hidden in plain sight. I love this phrase. I wish I'd come up with it, but I think it's a cryptographer's phrase. I think the microbes are visible to us as long as we know what we're looking at. They're almost like hidden text within another text. And we look for them by their field marks. We look for them by their manifestations, things that they do. And again, what they do is absolutely amazing. I also wish I had said this next thing. This is from an editorial in Nature. Thinking about us, thinking about our microbes, the new philosophical question is not who am I, but who are we? This also dovetails beautifully with Doug's talk this morning. Who are we? We are a human body with an enormous microbial community in and on it. And those microbes are doing some extraordinary things. Now I'm going to show you, we're going to get right down to the digestive system. Not with the slide that you're supposed to be reading there. I just want to show you that somebody has gone, not me, someone has gone and labeled this thing all up with what bacteria and what sorts of numbers we're finding in all different parts of our digestive system. You probably know this too, that we are packed with bacteria, and this is a favorite place for them to be, heavily involved with what we do for digestion. But now we're back to this amazing point. And bacteria can make neurotransmitters using ancient pathways. So what's a neurotransmitter? Neurotransmitter is a signal molecule that is going to be facilitating the passing of messages from one neuron to another. So it's pretty much how our brains and central nervous systems are working. It's the crucial molecule. But they're small and they're multi-purpose. Some of them are based on amino acids, and amino acids are just those things that all the proteins are made out of. Some of them are gases. Some of them are small amines, and I highlighted dopamine and serotonin because those are ones that you've heard of. But there's lots of them, all different sorts. They tend to be small, and that's crucial for my next point. Some people who are graduate students in biochemistry or chemistry might recognize a section of this. This back in the 70s and 80s and maybe into the 90s was the thing to have on your wall if you were a geek. This is the famous Boehringer-Mannheim chart of metabolic pathways. Again, you're not supposed to be able to read it even from the front row. The take-home story on this, and I'm going to do a little close-up of it for the next picture that will be meaningful, I think. The take-home story on this is that all the metabolic pathways are all interconnected in the most extraordinary ways. And of course, the biochemists are very proud about knowing a lot of these biochemical pathways. If you look at any section of this Boehringer-Mannheim chart, you see some amazing things, such as upper right-hand corner. Can you see dopaquinone? Dopaquinone. We're on our way to making dopamine. We're also on our way to making some pigments, but we won't deal with that today. That's going to be too complicated. But yeah, that's actually significant too. And look, we trace back from dopaquinone. In no time at all, we're in the realm of to some people anyway, familiar looking names for amino acids. There's tyrosine there, and it's hard to do this by looking at my screen. Here's another amino acid in here kicking around too, okay? My point is, these are some ancient pathways. I think the way to think about it is not, what are these bacteria doing making human neurotransmitters? Other way around. How did humans evolve to use these microbial molecules that have been made for billions of years for all sorts of other purposes that are part of these really intricate pathways and that are not owned by humans or mammals or anything like that? That's the key to how to ask the question. How simple does dopamine look? Simple, okay? You can almost make that an organic chemistry class. What does serotonin look like? Slightly more complicated. So I'm leaving you, I hope, convinced with the idea that yes, these neurotransmitters, these and others, can be made by microbes, are made by microbes, including the ones that are in our digestive system. Here comes our next picture. So what the take-home story from this one is that we have a two-way communication highway or pipeline between our digestive system and our brain called the vagus nerve, an ancient neuronal system. So that's what this is showing. There's essentially a highway of information. If we have bacteria in our digestive system making neurotransmitters, might some of them be affecting that nerve and sending signals straight to the brain, and might some signals be coming back from the brain? Could there be this two-way highway? And that, in fact, is the hypothesis about what is going on with bacteria in our digestive system that are making neurotransmitters. Now, I don't want to just leave this slide as is. Fabulous quote by Theodosius Dobzhansky, one of my favorites. So obvious it almost doesn't need to be said if you're a biologist, but nothing in biology makes sense except in light of evolution. So I show you this vagus nerve that's a highway between our brain and our gut bacteria, or at least that's the way it looks, and that's the way we're interpreting it today. But it doesn't just come out of nowhere. Now, what I'm going to say next could be a lecture unto itself, and it won't be, because I know I have a limited amount of time here. But I do want to point out that there's an evolution of the digestive system, too. Let's go to one of our really ancient ancestors, or in the lineage of our ancestors. Let's go to a jellyfish. kind of looks like a lampshade. The underneath part of the lampshade is essentially mostly its digestive system. It has some gonads in there too. But when you see that, you realize, of course it's got to have nervous system communication between its digestive system and whatever it has for a brain. And it doesn't have much of a brain, actually. But of course it's going to have that connection because its digestive system is essentially open to the environment. It makes perfect sense. So it's not like the vagus nerve popped out of nowhere. It comes from a long, long tradition of course you need to have some kind of communication between your digestive system and your whatever you're using for a brain, depending on which invertebrate you are. Okay? So now we get to slightly more heavy-duty stuff. We don't know much about the consequences of this. All the stuff I just told you, we don't know much about the consequences. And one more thing that I wish I had said, but the credit goes to Maureen O'Malley, who calls herself a microbial philosopher. She says we suffer from a residually macrobial perspective. I don't think she coined the word macrobial, but it's a great word. We're microbes, and we're rare. We're unusual on this planet. Most organisms are microbes. So if you go after this problem from a macrobial perspective, you're like, what's going on? The bacteria are making neurotransmitters. There are neurotransmitters, and we've got a vagus nerve. What's going on? No, you got to look at it from an evolutionary point of view. And you've got to look at it from a microbial point of view. Now, next picture, playground, is going to be my metaphor for how some experiments have been done to try to sort out that final question. What does it all mean? My friend who's a psychologist at Wheaton College said, Betsy, don't lead that audience into thinking that human children on a playground are equivalent to adult rats and mice. I said, okay, I won't do that. But I am going to use it in a very light kind of metaphorical way, because she's absolutely right. You have to be careful about some of these connections that get made. You could be a human observer of something called, I'm going to use the word anxiety because that's the word that's in the literature right now, anxiety. And you could say, no, you're using the word anxiety wrong, it's something else. But anxiety is, in fact, the word in the literature for this kind of study right now. You could go to that playground, you could be a human observer of child behavior, and you could say, that child is a little anxious. That child doesn't want to go out to the middle of the playground. That child is going to cling to me a little bit and not go toward the slide or the swings for some period of time. And this child is somewhat the opposite of anxious at this particular moment, right? So you as a human observer can determine what might be considered anxious and what might not be. We bring it to rodents, we bring it to mice, okay? We'll get to humans in a bit, but the initial experiments are on rodents. I got 2 mice. All I need to do is give one a placebo and one a probiotic, some dosage of live bacteria in some kind of a food or pill or whatever I'm doing with my dosing, okay? Lots and lots, dozens and dozens and dozens of experiments have been done on this. And there are a couple of caveats about it. One of them I think fits beautifully with this conference. You've got to know your animal. Mice are naturally a little bit anxious. They have to be. They're subject to predation. They tend to dash from one little covered spot to another. So you have to be very, very careful in these experiments about what you're actually looking at. It's not that mice should be complacent and lying out in the open, completely oblivious to any particular problems. So we're starting with an animal that's somewhat anxious to begin with. So I'm going to add another thing to our experiment. It works best, if you're going to get results for this, if the mouse is somewhat abnormally anxious, all right? Put the placebo, put in the probiotic with the other group of mice, and then you have to determine anxiety. And here's where my playground metaphor comes around. Think of that open field. This is a classic test that people do for animal behavior, but think of that open field as the playground. You've got a child who is dashing across the playground all different ways, the far left picture, or a child that's kind of clinging to the sides. And rodents generally going to kind of cling to the sides like they would in your kitchen if you were discovering one. They're not going to be dashing across the middle of the room probably. But this is essentially one of the tests that gets used. Is my mouse a little bit more anxious or a little bit less anxious? Okay? And another test also reminded me of a playground. Now, I know that as a rat. I'm going to get to rats in a second. It's a special kind of maze that has sort of like a playground aspect to it with some places where a rodent might peek out and go, gee, am I going to cross that little bridge or not? So another way to kind of determine whether that rodent is a little more outgoing or a little more anxious. Okay, so two of the many, many different tests that get done in order to determine anxiety in rodents. I did want to segue to rats because they're naturally a little bit more outgoing, so you've got to adjust all your expectations if you do this with rats. You're starting with a different baseline. And I'm making a big deal out of it because I am going to get to humans, and just think about what that baseline would have to look like to make this work with humans. Now, I'm going to stop here and say that what really helped me to prepare this talk is there are some meta-analyses of these studies. That means a separate set of researchers came in and took all of the dozens of papers, reanalyzed all the methods to make sure that they were spot on for the controls, reanalyzed all the data as though it were fresh data with a fresh set of statistics that you hope they did correctly, and came up with a conclusion. Here's the conclusion for rodents. If you start with a somewhat anxious rodent to begin with, it will become less anxious if you give it probiotics. Believe it or not, that's earth-shattering. Okay, so I'm saying that the conclusion is that the neurotransmitters being produced by the bacteria in the probiotics are contributing something in the way of neurotransmitters, and they can figure out which ones. There are more studies that we could talk about. We don't have time. In other words, it works. In other words, there's something going on. There's less anxiety in those mice and rats if you do the experiments right. So you go, okay, great, time to do it with humans. So there I am with a Siggy's yogurt cup getting ready for an experiment on myself. Here is the problem. There are a bunch of things that make me less anxious. The right amount of coffee, not too much, of course, glass of red wine, chocolate, cheese, comfort foods, reading a book. Actually, not reading a book makes me anxious. Reading calms me down. Family, Sunday mornings, playing basketball. So I am a very complicated human, and so is everyone else, right? So we're trying to do a human study here. We'd love to do that. I think the glass is half full on this. I'm going to end up concluding we don't know much about humans, but I'm going to tell you why we don't and why experiments could be done, and perhaps there will be some results at some point. Part of it is we got this great big bell curve. Of course, we got it for mice and rats too, but I think it's much, much wider for humans. You've got some human subjects. You're trying to figure out where they are to begin with on the anxiety scale. It's all about self-reporting, and it's extremely difficult to come up with what we would determine to be a so-called average person, that person that's sitting right in the middle of the bell curve there. But what you want to do, and which has not been done yet, because there was a meta-analysis on this topic for human studies, strangely, this has not been done yet. That's why the glass is half full. You need to start with anxious subjects. You might think, of course, that's obvious, but actually nobody's really quite done that yet. It's kind of hard with humans, right, to get a collection of humans that are all sort of similarly anxious about something. You also need control subjects who are also anxious if we're going with the whole anxiety thing. You need reasonable dosages. And I got to tell you, some of the rodent studies had some pretty heavy dosages of probiotics. Mm-hmm. There's that little thing, too, that you got to deal with. So somebody's got to really look at the dosages for this, okay? And then you need a few weeks of humans following directions, like, Betsy, don't have any red wine this week, no chocolate, no basketball, no reading. I want to see you at your most anxious. And then we'll see what all happens when you have the Ziggy's yogurt. So I'm going to say the human studies are hard. So my take-home story on this is not going to be, we're all set to go. We understand something about neurotransmitters and probiotics. But the glass is half full, in my opinion. So in conclusion, we still don't know much about the consequences of neurotransmitters produced by symbiotic bacteria of animals, but what we do know certainly is intriguing. Done.